Welcome everyone to our first State of the Science snapshot from the Science and Technology team at the Good Food Institute. My name is Liz Specht. I'm the VP of Science and Technology, and I'll be joined today by several of my colleagues to highlight the most exciting developments happening in the science and technology ecosystem for alternative proteins. These snapshots will be presented 3 times a year, and each will cover developments over the prior 4-month period. First, I'd like to give a brief rationale for why we've launched this snapshot series. While we include some technical insights in our flagship annual publications, such as the State of the Industry reports, alongside the annual trends in the private sector, such as investment and company launches, we've noted that things are moving really quickly in the research and development space, and that will only get faster as more resources start pouring into the alternative protein sector. I put up this graph just to illustrate the exponentially compounding nature of research in this space. These bars reflect the number of publications just from GFI's grantees over 4-month cycles throughout the year. And this represents, of course, obviously a narrow but pretty representative slice of the overall research happening in this space. And you can see there's a huge increase even just from one 4-month period to the next. So while that annual cadence of things like the State of the Industry reports is great for a comprehensive look at the sector, we wanted to provide some insights on exciting trends that we're catching or new important papers being published in between those annual updates. While it of course takes time for these technology advances to translate into true commercial impact, our intent is to really use these snapshots to keep stakeholders informed about the frontier of knowledge in this field. And these snapshots are intended to be just that, a single snapshot in time. These are not intended to be fully comprehensive of everything happening in these 4-month periods, but rather to provide some more curated highlights of the things that we're most excited about. In some cases, we actually don't even have specific publications or announcements to point to, but rather our subject matter experts are simply picking up on signals from conversations we're having or glimpses we're getting to at some pre-published work that we can aggregate into larger trends that we're seeing. So today we'll cover not just the technology itself, but also touch on developments in the broader scientific ecosystem, including noteworthy funding announcements and updates regarding the growth of the talent streams leading into this field, since these funding and talent considerations are 2 of the biggest bottlenecks at the top of the funnel. As more talent and research funding flow into the alternative protein sector, all of these advances will pick up steam. So first, we'll hand over to Dr. Priera Panescu to talk through developments in the plant-based space. Priera, take it from here.
Excellent. Thank you so much, Liz, for that introduction. I'm excited to kick us off with the technical developments for the first 4 months of 2022 for plant-based proteins. Next slide. The first trend I'll speak to is the application of upcycled ingredients to create novel functional plant-based ingredients. The first company I will point to is Leafed. They're a New Zealand company that just closed $15 million funding round to develop protein extraction methods from leaves to get rubisco protein from them. So rubisco protein is one of the most abundant proteins in the world because it's pretty much found in everything green. It's required for photosynthesis. So really being able to leverage extraction techniques to get this abundant and very high value protein is really exciting. And we're really excited to see what Leaf gets up to. DSM also announced they are making an upcycled canola protein isolate called Canola Pro in commercial quantities in a new facility in France. And other companies like More Foods, they're focused on upcycling pumpkin and sunflower seed solids to make textured plant-based meat, and Crush Dynamics, which is using upcycled grape derivatives from wine waste to create nutritious plant-based foods. There are also a plethora of academic articles focused on extraction from leaves as well as other upcycled ingredients. Here are just a few of the articles highlighted that are working on traditional wet fractionation in order to get proteins from leaves, all the way to using more advanced biochemical and physical methods, and then actually testing the functionalities of these extracts. The paper on top here is actually from a GFI grantee, Ophir Benjamin. Next slide, please. The next trend I would like to talk about are the development of plant-based fat technologies and the continued use of emulsification and encapsulation to solidify plant-based ingredients. Givaudan developed a technology called Prime Lock Plus that they claim can reduce the fat content of final products by up to 75%. The technology mixes flavors in coconut oil separate from proteins and then mixes them together later. The coconut oil and the flavors are encapsulated so they don't leach as quickly when cooking. Lipid secured a $4 million fund to scale up their phytofat platform, which uses encapsulated plant oils like sunflower oil or olive oil, as well as gums and fibers. All generally regarded as safe, and they plan to create up to 10 tons of phytofat per year. Good Food Technologies is another company working on proprietary fat technology, and they call it Aromex, and it uses a konjac, which is a root plant, to mimic pork fat. And finally, GFI grantee Professor McClemens also developed a plant-based adipose tissue using a high internal phase emulsion and emulsion gels. So the common theme here is that plant lipids, although they're usually oils at room temperature, can be structured so that they can mimic animal fats as well. Next slide, please. Excellent. And then the next trend we saw are more advances in texturization. So I'll first start with extrusion. 2022 is kind of emerging with extrusion 2.0. So first I'll talk about Martin Hoffman, who is launching a spinoff from ETH Zurich using a new unique extrusion technique. So he envisions this extrusion technique in the product to be a B2B ingredient. And essentially what it does is it has two extrusion dyes. One is with pea protein and the other is with fat, and they feed into each other into one extrusion line that is then mixed using a software that's proprietary. This is going to allow for marbled plant-based meats without the need for 3D printing. Food Plant Facility is also being launched in Singapore, and they have small scaled machinery such as high moisture extrusion to help with smaller companies being able to test out their products at smaller scales. And finally, ADM plans to double their extrusion technology this year in Illinois. Excellent. The next update in texturization is in 3D printing for whole cut mimics for plant-based and hybrid meats. So Alt Farm is an example of a company using 3D technology to 3D print pea, soy, and algae protein for a Wagyu beef. Plantish has developed a 3D printed whole cut salmon fillet from legumes and algae extract. And Nova Meat launched the ultimate hybrid 3D product, which contains cultivated animal meat, animal cells, plant-based derivatives, fungi, algae, and spirulina. And it's a blue steak, which gives a futuristic look to the plant-based meat. It's very interesting seeing all of these hybrids together. Next slide, please. Excellent. And the final update I'll give on texturization is so we're actually seeing these really great fiber spinning innovations. So Tender is an example of this. They're using a rotary jet spinning technology, which mixes soy proteins, dietary fibers, and other carbohydrates to create hair width fibers organized like fibrous muscle tissues. So right now their current capacity is several pounds per hour, but they're looking to pilot test at restaurants and formally launch next year. And this technique was actually originally developed for cultivated meat scaffolding. And they did receive a grant from NSF to develop the scaffolding technique for cell cultivated meat as well. But for Tender, it's being applied in a plant-based way. Next slide, please. And then I'll give some final updates. There is a new journal, Future Foods, which is completely open access, and it has a lot of really great alternative protein academic articles. Highly recommend it. Kellogg partnered with Benson Hill to make their soy Morningstar Farms products more sustainable using their ultra-high soybean variety. Umaro Foods, a creator of seaweed-based bacon and a GFI grantee alum, received support of $3 million from Ag Funder. And this was just announced right before they appeared on a Shark Tank episode where they received $1 million for 7% equity from Mark Cuban after only asking for $500,000 for 2% equity. Additionally, Colombian Future Seeds launched, and it's the biggest tropical seed bank in the world. It includes the largest collection of bean and cassava germplasms, so it could be really strong for alternative proteins. Australian Plant Proteins, with the funding of the government, invested $268 million in USD to advance the construction of 3 new production plants to quadruple animal-free protein production capacity in South Australia. And finally, the Danish government launched a $100 million USD fund called the Plant Fund to specifically invest into plant-based foods. Thank you so much. And with that, I would love to pass it on back to Dr. Liz Specht.
Thanks so much, Pri. First, I'd like to highlight this publication that came out 2 months ago from a collaborative set of authors between Colorado-based fermentation company Mycotechnology and researchers at Cornell and University of Illinois. This is a great example of what we see as really a growing trend to leverage fermentation to improve the functional properties of plant proteins, including things like improving digestibility, while also reducing, for example, undesirable off flavors. This approach is actually already being used commercially in commercially available products through a partnership between Mycotechnology and Plantera Foods under their Ozo brand. So it's really great to see private sector players providing more public data about their proprietary approaches through these academic collaborations to get that data out in the public domain for others to build upon. The next area I'd like to highlight is a growing interest in using feedstocks other than pure sugar for feeding the microorganisms used in fermentation for alternative protein applications. So by leveraging feedstocks that are more crude or less well-defined, such as side streams that result from various agricultural and food processing facilities, can improve both sustainability and cost effectiveness of alternative protein products. This particular paper from a research group in Sweden demonstrated using a side stream from potato starch production at demonstration scale. So this included scaling the process all the way up to a 1,200 liter airlift reactor. I just want to point out these demo scale data points are incredibly valuable for validating new processing methods and feedstocks to show that they can actually work beyond bench scale, where some of the practical considerations like proper mixing or impurities might be harder to detect and therefore might not give folks a glimpse at what challenges may arise at commercial scale. So it's really great to see these academic groups increasingly including scale-up data in their investigations and their papers so that new technologies like this can be more readily adopted by commercial players rather than getting stuck in the so-called valley of death between academic research and commercial use. In the first few months of this year, we've seen a number of great review articles published on the use of fermentation in the alternative protein space, which I really recommend for a comprehensive overview of the latest in the field. These 3 articles in particular have a detailed mention of alternative protein use cases for fermentation. One reason I was especially excited to see 2 of these articles was their focus on using fermentation to produce smaller peptides and hydrolysates. Hydrolysates are essentially chopped up bits of larger proteins, and these can be a key component of the cell culture media for cultivated meat production. In fact, using plant protein hydrolysates in place of individual purified amino acids has been proposed in several of the techno-economic analyses that have been published as a key component for reducing the cost of production of cultivated meat. Mild hydrolysis can also, in many cases, improve the properties of plant proteins, such as their solubility. So it's great to see these compilations of a number of different studies that are using fermentation to hydrolyze proteins with an eye towards achieving specific types of properties or ideal target sizes for those protein fragments. The final area I'd like to highlight is more of a recurring theme that we're starting to see more attention being paid to in our various conversations with both academic and private sector researchers. So Priera mentioned a lot of focus on hybrid products across the three main alternative protein production pillars. So between plant-based, fermentation, and cultivated meat. But we're also increasingly starting to hear more interest in and exploration of this hybrid mindset as applied to the categories within fermentation too. So in most cases, we think of fermentation as playing mostly one of these three roles. Either it's converting a substrate into a more desirable form, that's traditional fermentation, or we're using it to grow the whole cell biomass of the microbes, but the feedstock is being removed, so that's biomass fermentation. Or the microbes are simply being used as production hosts for a specific high-value ingredient, which is then purified away from the rest of the cells and the feedstock. But increasingly, we're hearing interest in folks looking to leverage all of these components, the feedstock, the whole cell biomass, and specific ingredients that the cells can be selected or engineered to produce. And this sort of hybrid mindset can really reduce downstream processing costs associated with separating each of these components and then recombining them in the final formulation. So I don't have specific companies or research papers to point out on this just yet, but I wanted to flag this as a trend that we're starting to hear more about in some of our early stage conversations. So with that, I'll hand over to Dr. Claire Baumkamp.
Thanks, Liz. So there is too much, honestly, to talk about in cultivated meat to do it in 5 minutes, but I will try to give you a couple of quick highlights. So the first paper I wanted to talk about was this preprint by Venkatesan et al. 2022, where they were looking at recombinant growth factors from a variety of different species and producing them cost effectively and then comparing their efficacy in cell culture. So what this first graph is showing is proliferation rates of cells in culture that are grown with FGF2 that's recombinantly produced from a variety of different species. And then on the right is the commercially available FGF2 as a control. And so what you can see is that for several of these species, including salmon, tilapia, and medaka, you actually see either equivalent or better functionality as evidenced by higher proliferation compared to commercial FGF2. And what's really exciting here is not just the proliferation, but that this is being done cost effectively. So if you could advance the slide, Renee. So if we compare to the commercially available forms of both TGF-beta, FGF2, IGF-1, and PDGF-BB, so these are quite pricey if you try to buy them commercially. So whether from R&D Systems or PepperTech, we're talking about, $300 to $7,000 for a milligram of protein versus in-house production, as they estimated in this paper, was about 10 or 20 Canadian dollars per milligram. And so that's a big advance over the commercially available products. So lots of low-hanging fruit in terms of bringing those costs down. And if you could advance the slide a couple times. So what this translates to in terms of, the cost in the media. So for TGF-beta and FGF2, which are some of the main cost drivers in cultivated meat, serum-free media, we're talking about a 720-fold and a 255-fold decrease in cost. And then when you translate that into the cost of the full media formulation, that becomes an 11x decrease. So pretty substantial. And we're going from most of the cost being driven by the growth factors to most of it being driven by the basal media. So this research was carried out by a team that included Dr. Peter Stogios, a GFI grantee, and he actually gave a webinar quite recently talking about some of this work. So you can find that on GFI's YouTube channel, as well as another webinar by Dr. Cameron Semper, who is another member of the research team, talking about where they were at with this project last summer. So check out both of those. And then the next paper I wanted to touch on was Zernov et al.'s work on edible microcarriers. So what they were looking at here was making microcarriers from collagen and chitosan, with the idea that if we can make something that's edible, you don't have to do the step of removing the microcarriers from the cells, which can, make the bioprocess more complicated. You're introducing more reagents, just basically increasing your costs. And so they found pretty good results in terms of cell adhesion with the collagen microcarriers, but not so much with the chitosan. And so the challenge here is that you could produce chitosan from fungi, and that's great. For collagen, you're talking about either animal-derived collagen, which is not really compatible with cultivated meat, or you're talking about recombinant collagen, which is quite pricey. So what they did next was they combined collagen and chitosan in a 90-10 ratio, so 90% chitosan, 10% collagen. And they found good adhesion under these conditions and good proliferation, both for mouse cells as well as for rabbit, sheep, and cow. And so that's pretty exciting in terms of being able to scale up cultivated meat production. And so this research was carried out by a team led by Dr. Marcel Machlouf, who's a GFI grantee. Next slide. And so I just wanted to quickly highlight this paper by Zhu et al., where they were looking at nutrition in terms of amino acid profiles as well as taste, and comparing cultured cells with conventional meat. So this was satellite cells from chicken and cow. They're proliferating and differentiating to make basically a very simple cultivated meat prototype. And then using an electronic tongue system to look at various taste compounds in culture, or in these cultured cells, and comparing those to conventionally produced meat. And so they do find some differences between their samples and the conventional meat. But I think what's really exciting about this study is that it's really sort of laying the groundwork for this kind of comparison being done. And so I think we'll see a lot more experiments like this as researchers continue to produce cultivated meat prototypes in their labs. And this will really give us a lot more insight into kind of what needs to be done with culture conditions to make cultivated meat that tastes like conventional, or even better. And so on the next slide, I just wanted to highlight a quote from this review paper out of the Biosense Institute, where they talk about the need for open science and sort of being not locking everything up as intellectual property, and how that's going to really be like the rising tide that lifts all boats for cultivated meat. And so one thing that kind of gives me hope on this front, on the next slide, is publications from cultivated meat startups. And so we've seen Aleph Farms, Biotech Foods, and actually 3 papers from Mosa Meat, where companies, I think, are really recognizing the need to move the whole field forward. And so this is, I think, a really great sign for the future of this industry, and, a true spirit of collaboration. And so on the next slide, I'm going to transition over to talking about developments in alternative seafood. So the first paper I wanted to talk about by Zhang et al. Was one where they made plant-based scallop prototypes from pea protein and pectin. So they're mixing those together, doing some fancy phase separation related chemistry, and making this nice scallop prototype. And so in the photos they presented in the paper, you can actually see the grilled scallop analog compared to a conventional grilled scallop. And they look pretty similar. And then they also did some textural analysis of the grilled conventional and plant-based scallops. So comparing things like hardness, cohesion, springiness, and chewiness. And so they do see pretty good correspondence in terms of those textural measures between the plant-based and conventional scallop. And this research was carried out by a team that included Dr. David Julian McClements, who Priya already mentioned. And so on the next slide, I wanted to talk about this paper because I think it has the potential to be really impactful for cultivated seafood. So what they did here was they isolated a fibroblast-like line from discarded fin tissue from Filefish. And what they found with this line was that it could actually be differentiated pretty easily just by changing the culture conditions. And so this is a little bit different from our usual intuition about terrestrial cells, where, you know, to transdifferentiate a fibroblast into a muscle cell, you might have to do something like expressing the MyoD gene. So then we're talking about genetic modification. But if it's truly the case that it's easier in fish cells, that could really open the door for you know, a lot of different approaches, and could make the challenge of getting appropriate cell lines for cultivated seafood, it could make that a lot easier. And so I was pretty excited to see this paper, and I'll be excited to see if this is sort of a more broadly applicable finding, or if this is just a particularly cool cell line. And then I wanted to quickly point out this review paper by Goswami et al., who's Dr. Goswami, the lead author on this, is a GFI grantee. And so this paper was on various uses of fish cell lines in research, including in cultivated seafood. And then I also wanted to point out this paper, which is actually the first publication from the Algae to Fish project, which GFI is funding, led by Dr. Federico Ferreira. And so what they did in this study is they're basically validating their carrageenan-based bioinks, which are derived from algae. And so they're first growing mouse fibroblasts on scaffolds made of this carrageenan-based material. And they find that, you know, with some optimization, they grow pretty well. And then they also demonstrate bioprinting using this same material. And so this is the first phase where they're just validating with mouse cells, but then the eventual goal of this project is to include electrospun fibers, which will help with myofiber alignment, and also to grow sea bass cells on these same scaffolds. And speaking of algae, I just wanted to quickly highlight a couple of other papers that have come out. So here we're seeing Dr. McClements again. So this was a study where they looked at off flavors in algal oils, which are rich in omega-3s and obviously important inputs for both plant-based and cultivated seafood, and trying to kind of find methods for reducing those off flavors. Next, there was a study where they looked at using microalgae to impart flavor into plant-based seafood, which is obviously a key need. And then finally, there was a review on fluorescence spectroscopy as a monitoring technique for basically scaling up microalgae production. So I think that'll be a really key enabling technology that will allow alternative seafood broadly to succeed. And so with that, I will turn it over to Amy Huang to talk about the talent and training ecosystem.
Thank you so much, Claire. 2022 is off to a rocking start in the way of education and training advancements. On the next slide, you can see that we've seen a steady uptick in both the number of alternative protein courses being offered by universities around the world, many driven by GFI's Alt Protein project. And we've also seen a skyrocketing demand for these types of skill building opportunities. You can see our course database to really take a deep dive into all of the courses that are on our radar. In addition to these kinds of individual alternative protein courses, we're also starting to see alternative protein programs appear on the scene, which is a super exciting development. Tufts University officially launched its minor in cellular agriculture, which is a hugely exciting step in the way of bolstering the legitimacy of the alternative protein field as a field of study by allowing students to really fully specialize their academic careers on alternative proteins. And on the next slide, you can see that student demand is really on the rise. One data point that signals this overarching trend line is the number of registrants for GFI's free online course on alternative protein science. As of the end of April, we have over 9,000 registrants, up 1,000 since just the end of 2021. And the fact that students are driving forward many of the educational pathways is also a really strong signal that student demand for alternative protein focused training opportunities is on the rise. Just a few highlights from these student-driven initiatives. In January, the Cambridge Alt Protein Society launched the Alternative Protein Fundamentals program, which remotely educated over 400 students, scientists, policymakers, and entrepreneurs on advancements and opportunities in alternative proteins over the span of 8 weeks. In March, Stanford University launched the second iteration of their Alt Protein Project-led course on rethinking meat, with over 300 registrants this year, up from 100 in 2021. And in May, the UNC Chapel Hill Alt Protein Project wrapped up the university's very first course on the science of cultivated meat, led by professors Doug Van Steele, Andrew Gladden, and Alan Jones. The Chapel Hill Alt Protein Project is expecting this course to become a permanent fixture in the biology department, to be taught year over year by faculty as an integral part of the university's curriculum. And excitingly, we expect this trend to only continue as more and more students join the alternative protein movement. In addition to these developments, we're also beginning to see the emergence of new private sector solutions focused on helping the alternative protein sector overcome its talent bottlenecks, with sector-specific recruiting and training organizations like Talus appearing on the scene. And this is also a super powerful signal of the increasing maturity of our field, given that there are lots of demonstrated business models from established fields for these types of talent-focused solutions and services. So very excited for the 4 months that we have ahead, and can't wait to circle back on some of the developments that are underway. With that, I will pass it over to Kyle Sosi, GFI's Research Grants Program Manager.
Thank you so much, Amy. And I share all of my colleagues' enthusiasm and excitement for the first few months of 2020. For our research funding, the first 4 months of 2022 have brought exciting announcements of significant and historic alt-protein research funding from governments around the world, as well as the announcement of new funding opportunities from GFI. Though we're only a third of the way into this year, GFI has already released our 2022 request for proposals, which will award $3.9 million in new funding. Past GFI grantees have received millions of dollars in new funding from government sources around the world. And the Netherlands announced the world's largest ever public investment in the cellular agriculture field. Let's go to the next slide. Through GFI's research grant program, we continue to support high impact open access research into alternative proteins globally. We've released our 2022 request for proposals this April with a submission deadline of June 3rd. This RFP will award $3.9 million in new funding to projects focused on pressing scientific and technological challenges facing the alternative protein industry. Though we are allowing general applications, this RFP is primarily focused on 3 core research focus areas, those being plant protein processing, albumin and transferrin replacements, and alternative seafood flavorings. Now transitioning to external funding announcements we've seen so far this year. Governments around the world have stepped up to increase their investments in alternative proteins, in many cases with GFI researchers and grantees involved, or with the funding catalyzed by advocacy from GFI. So the United States Department of Agriculture announced $2.7 million in funding for 5 alternative protein projects. And importantly, 3 of the 5 funded projects are led by current or former GFI grantees. Washington State University received $595,000 for plant-based meat research. UMass Amherst received $596,000 for plant-based meat research. And UCLA, under the direction of Dr. Amy Rowett, received $604,000 for cultivated meat research. Dr. Amy Rowett's team was also successful in receiving an additional 5-year, $1 million grant from the National Science Foundation to grow her cultivated meat research program. Finally, and possibly most excitingly, in Europe, the Netherlands announced a 60 million euro or $65.4 million investment for cultivated meat and precision fermentation. This is the world's largest ever public investment in the cellular agriculture field. And this historic investment in cell ag is from the country's National Growth Fund, which is a 20 billion euro effort launched in 2022 to invest in projects that generate sustainable economic growth for the Netherlands in the long term. These examples just barely scratch the surface of the new funding activity in alternative proteins, and we are so excited and looking forward to what the rest of 2022 brings. And with that, I'd love to pass it back to Dr. Liz Specht to close us out.
Fantastic. Thanks so much, Kyle. That wraps our first State of the Science Snapshot covering January through April of 2022. And we're excited to bring these snapshots to you every 4 months throughout the rest of the year. As always, in the meantime, you can keep up to date with all of our resources and publications by following us on social media and enrolling in our newsletters. And if you'd like a deeper dive on the science at any time, check out our detailed explainers, which also link to lots of additional research papers and resources. Thank you for tuning in.